much, Rutledge. That was really a tour de force. And I'm sure there will be quite a few questions during the breakout on the next step of application into human clinics. Really fantastic work. Well, I'm pleased to continue to build on the outstanding presentations during this session with our next speaker, Dr. Alexey Krasnoslobodtsev. And Dr. Krasnoslobodtsev is a doctorate in physical chemistry with a master's in chemistry and a bachelor's degree from the Novosibirsk State University in Russia. His PhD and master's degrees were earned from New Mexico State University. Dr. Krasnoslobodtsev is currently a research associate at the University of Nebraska Medical Center, where his research interests are focused on the application of single molecule methods in studying protein misfolding phenomena as well as self-aggregation. Dr. Krasnoslobodtsev's talk today is entitled AFM Force Spectroscopy as a Nanotool for Early Detection of Misfolded Protein. Clearly a very important need, a very important application of nanotechnology. Please join me in offering a warm welcome for Dr. Krasnoslobodtsev.
Well, I would like to thank the organizers for giving me the opportunity to talk today at this very nice conference. And to be honest with you, I'm a little intimidated by the conference because I'm not clearly a distinguished professor or scientist. I'm a recent graduate, graduated in 2005. But I'd like to use this opportunity to talk to you about what I have been doing for the past 2.5 years. And I'll be talking about using force spectroscopy as a nanotool for early detection of misfolded proteins. And my presentation will be of general nature, but I'll try to keep you as entertained as you were by previous speakers. So if you could take one sentence home, and this sentence is, single molecule force spectroscopy can be used to do good, can be used to detect misfolded conformations of a protein that will go ahead and aggregate. And also it can be used to identify effective therapeutics to prevent this aggregation. Then you don't have to listen to my presentation, all right? So the outline of my presentation is here. I'll give you a short introduction into misfolded phenomenon as it is related to conformational diseases. And I'll talk about single molecule approaches, which is force spectroscopy to study misfolded phenomenon, talk about its advantages and applications. And at the end, I'll touch dynamic force spectroscopy, which what I call is an area beyond measuring forces of intermolecular interactions. So, as we all know, for functional activity of a protein has to be folded in a certain conformation. And where's the, oh, here. And natively folded protein can assume misfolded conformation which is induced by certain stress. And this stress can be environmental, which is elevated temperature or shifts in pH, for example, osmotic stress, osmotic shock, and also some other kind of stresses could induce the misfolding of the protein. And also undesirable mutation can cause misfolding of the protein. So if a protein is misfolded, then the misfolded conformation of this protein is prone to aggregation. So the misfolded protein can be, to some extent, risked by chaperones, which are distinct class of proteins. But let's assume that we have a lot of stress, or chaperones were busy, and then the misfolded protein will go ahead and aggregate. All right? So when it aggregates, it forms small oligomers or protein fibrils, and then we deal with pathological conditions or a disease. Okay. Nowadays, many human diseases are recognized as conformational diseases. In essence, this is assumption of protein. The protein assumes misfolded conformation that aggregates. All right. And this picture shows some of the examples. For example, plaques and tangles in Alzheimer's disease. And these are deposition of misfolded proteins. Lewy bodies in Parkinson's disease, intranuclear inclusions in Huntington's disease, and plaques of prion amyloid. And also amyotrophic lateral sclerosis aggregates. But when we look at these aggregates, you can see that phenotypically they're different. However, they all share a common link. And the common link here is that these depositions are aggregates of misfolded proteins, and these misfolded proteins are prone to aggregation. And also, these aggregates cause molecular stress and interfere with with cellular function. So, in the mechanism of aggregation, the stress, let's say environmental stress, induces misfolded conformation, and this misfolded conformation is aggregation prone, and we can call it sticky. And this misfolded protein interacts with normally folded protein. It causes misfolding of that protein and the assembly of the two. Okay, and then this cycle multiplies the copies of misfolded diseases, misfolded proteins, and ultimately it leads to aggregation, formation of oligomers and large aggregates and fibrils. We have set a goal for ourselves to use single molecule methods to look at this step here, which is dimerization of misfolded protein. And this is the first step in the long pathway of aggregation. And you may ask a question, why would we want to look at the dimerization? And I can tell you why. Despite a large number of experimental data, huge wealth of research that has been done on aggregation, there's still no consensus on whether small oligomers or large aggregates or fibrils cause toxicity. So what we want to do, we want to address the very first step in this aggregation process, and perhaps we'll be able to stop aggregation at this first step. So this slide shows a current view of possible approaches that can be used to therapeutically intervene in this aggregation process. And along the aggregation process in which native protein misfolds and misfolded monomers oligomerize, and then they go ahead and form fibrils. Each step of this aggregation process can be, at each step we can intervene therapeutically and prevent it However, these two steps, namely clearance of the fibrils and inhibition of fibril formation, can be very dangerous. Because as I mentioned before, we don't really know what causes the toxicity of the toxicity and the diseases. And therefore, the clearance of the fibrils and the inhibition of fibril formation can lead to accumulation of small oligomers that, in fact, may be the most toxic species. So we cross out these steps, and while all these possible therapeutic interventions look good, I'll focus on these two, which is inhibit of oligomerization and stabilizing monomer or upregulates chaperone activity. And in fact, these two steps could be the easiest ones. And they can be done by introducing small molecules that can bind to specific regions of the misfolded protein and stabilize it. And these molecules are called chemical or pharmacological chaperones. And some of the examples are osmolytes. For example, natural osmolyte proline was shown to inhibit aggregation of a protein. So it can be done. And I'll show all of it. But despite the crucial importance of protein misfolding phenomenon and these abnormal interactions, very little is currently known about the molecular mechanism underlying misfolding process. And the reason is because all of the methods that are out there and available, they do not address the early dynamic steps in the aggregation process. But what is available out there to help us address these issues? And what we have is single molecule atomic force microscopy, force spectroscopy, to address the early stages of aggregation. And therefore, we propose this method for identification and characterization of misfolded aggregation, as well as we think that it should help to identify rational therapeutics. So what can single molecule force spectroscopy offer? As I mentioned before, we want to address dimerization of misfolded proteins, and apparently in misfolded state, the proteins interact to greater extent compared to normally folded proteins. And what we do in a single molecule force spectroscopy, we covalently attach molecules to sharp AFM tip and the surface, and then we probe the surface. When tip approaches to the surface, there is a chance for molecules to interact. And then when we withdraw tip from the surface, we break the newly formed complex, and that produces characteristic force curve. So FM force spectroscopy allows studying binding strength of intermolecular interactions. And in order to know what kind of information we can obtain from such measurements, one has to know the anatomy of force curve. I'm not going to go into details, not to bore you to death, but I'll have to mention that we're using long flexible linkers to move interactions far away from the surface, and therefore we can unambiguously identify this specific interaction. And also, the most important point is this point right here, which is rupture point, at which we rupture formed bond. And go back. And by measuring the jump in the force, we can measure the specific force which is required to break the specific interactions formed between molecules. So I'm going to skip this. No, no, I'm not. So in my experiments, I have used short peptide, 7 amino acid from sub-35 East prion, and this peptide has been shown to aggregate, and the fibrils that this peptide forms resemble very well the aggregates that are formed from full-length protein. And unless otherwise specified, I'll be talking about experiments related to this peptide. So I'll show all of it. So we have done some experiments with this peptide at different pH values. And this pH value is 2, 3, well, that's not the point. The point is that the morphology of aggregates is different at different pH values. All right. At pH 2, for example, the peptide forms fibrils, and at pH 3.7, it forms small globular aggregates. At pH 5.6, there are some small aggregates, but mainly what you could see is amorphous material on the background. So when we went ahead and measured forces of interactions at these conditions too, we saw that at low pH value, the forces are higher than at other pH values. All right. So if we link this to morphology of aggregates, then we can say that the conditions that induce misfolded conformation that has high propensity to aggregate also produce high forces. So once we have identified that aggregation propensity can be detected by measuring forces of interactions, we envisioned, I'm going to skip this. We envisioned a device that could be used to identify effective therapeutics. And this device is based on AFM for spectroscopy. Here you have AFM tip with covalently linked protein and probed surface with the protein, and you probe interactions in continuous manner. And what you measure is forces of interactions. So if you want to test therapeutical agents, say drugs, for this particular set of drugs, the drug number 2 would be the most effective, and it would be the best candidate for further development of effective therapeutics, just because it reduces forces of interaction. But of course, what we want to see is not the reduction of the forces, we want to see zero forces, right? And there are some challenges which have to be overcome in order to use such device. One of them is surface chemistry. Has to be stable enough to allow continuous probing of the interactions. And we have recently introduced a new chemistry which is based on silatran, which is similar to silent chemistry, but it can be done in aqueous solutions, and therefore it can be used for immobilization of biomolecules in one step. And also automated exchange of buffers containing drugs of interest and automated data analysis would facilitate the use of such device. So in the next few slides, I'll talk about dynamic force spectroscopy. And in dynamic force spectroscopy, the interactions are probed over a wide range of loading rates. In a simple description, the force is related to loading rate through this equation, and the dependence is logarithmic. So if we, the animation doesn't work. But anyway, in case number 1, we pull on our dimer slowly. And in case number 2, we pull fast. And the forces would be different. For case number 1, the force is smaller than for the fast pulling. So what one can obtain from such measurements is that one can access the energy landscape for dissociation reaction. And you can see the dependence of rupture force on the logarithm of loading rate. And what you can measure is, first of all, the distance to the transition state from the slope of this regime. And the distance is, in the energy diagram, is the distance from the bound state from the bound state to the intermediate. And what you can also do is you can measure dissociation rate constant, which is thermal off rate for the dissociation reaction. And here I show the force distributions measured at different loading rates. And you can see that force does indeed increase. So this slide shows the dynamic force spectrum that was obtained for this short peptide at pH 2. And as you can see, it reveals two distinguishable linear regimes. And the number of linear regimes is usually attributed to the dissociation of a complex that involves overcoming of more than one activation barrier. And this schematic shows that we have two energy barriers on the dissociation pathways. And the estimated positions of inner and outer barriers are 0.2 and 3.5 angstroms. And the off rates were estimated to be 286 and 0.9 reciprocal seconds. So what we can obtain from these measurements is the lifetime of a dimer for this peptide, and the estimated lifetime is 1.1 seconds. So you can imagine the entire process as peptides that have conformational dynamics, and the dynamics is in nanosecond, microsecond scale. So when they come together, they associate, right? And they get frozen for 1.1 seconds. And if you compare it to dynamics of monomers, that's 6 orders of magnitude increase. So it gives a chance for this aggregation to happen once the dimer is formed. And this potentially could provide a possible explanation for the phenomenon of the protein self-assembly. And in summary, I would like to repeat the first sentence that I said, that we think that single molecule force spectroscopy can be used to detect misfolded protein that is prone to aggregation, because there may be some other misfolded proteins that are not prone to aggregation. And also it can help identify the effective therapeutic agents that could prevent aggregation at very early stages of the aggregation process. And also I'd like to say that the stabilization of a dimer by 6 orders of magnitude can be an explanation. Basically, the dimer has a crucial role for the aggregation to proceed. And I'd like to acknowledge Dr. Yuri Lubchenko, who's the head of my lab, and the lab members. And I would like to thank all of you for your attention.